Sequence Reads Tool, Genomic Data Commons, GDC 2.0 Video Guides, Bill Wysocki, PhD, GDC User Services Lead, Center for Translational Data Science, University of Chicago. The GDC Data Portal 2.0 Sequence Reads tool allows users to visualize BAM files that have been harmonized by the GDC that are associated with their cohort. In this GDC video tutorial, we will visualize sequence data for our active cohort, which was previously created with lung cancer patients who were 50 years of age or older at diagnosis. For information on creating a cohort, see the Cohort Builder tutorial video. Starting at the Data Portal homepage, portal.gdc.cancer.gov, the Sequence Reads tool can be reached by clicking on the Analysis Center icon at the top left of the page. Within the Analysis Center, the Sequence Reads tool can be found in the card second from the right and can be opened by clicking the play button within the card. Note that the card may be in a different position based on your screen size. Using the Sequence Reads tool. Before we start, we would like to remind you that the data shown in the Sequence Reads tool is controlled access, and that any unauthorized display or sharing of these visualizations and data may be in violation of the data use agreements for dbGaP projects. In this demonstration, only open access data visualizations will be shown, which are not available in the GDC at this time. When we first enter the Sequence Reads tool, we are met with an access alert message that prompts us to log in. Once we do log in, we are prompted to enter a search string. This can be a file name, file ID, case ID, or case UUID. Alternatively, we will choose the button below the search field, which will display the first 1000 BAM files in the cohort. When we click on this, each BAM file will be displayed along with its associated case, sample type, experimental strategy, and file size in gigabytes. We choose the first one in the list, which is an RNA-seq BAM. When the BAM file is verified, which we can see based on the green check mark, the entity ID and aliquot barcode is displayed along with the experimental strategy, sample type, and size. At this point, we either select a variant or enter a gene or region in the field. We will enter TP53, which will convert to chromosomal coordinates when the gene is verified with another green check mark. Other formats are accepted and example input is listed below the field. We now click on Submit, which is located at the bottom. Main visualization screen. The top section of the Sequence Reads tool shows the different gene transcripts and coordinates based on the GenCode 36 gene model, which is used by the GDC. Below the transcripts is a coverage graph that describes the coverage at each region. Below the coverage graph are a graphical representation of the reads as they are aligned on the chromosome. When zoomed out, they appear as small bars, but their sequence can be read when zoomed in. Changing the view. The region that is visualized can be changed by changing the coordinates in the field at the top left and pressing Enter. Here we will change it to coordinates with an even 1000, which changes the view and zoom accordingly. To maintain the same level of zoom, but browse up and downstream the chromosome, click and drag the visualization left and right. To zoom in or out of our current region, we can use the in or out buttons, which are available as ×2, ×10, or ×50. Each part of the graph can be hidden or reappear by choosing Tracks and GenCode to remove the gene markers or the aliquot name to remove the read information. Choosing the Config button at the right side of the gene markers or reads brings up a panel that allows us to change visualization properties. Additionally, this panel displays a legend that indicates what each read color represents. A legend for the GenCode genes can also be found at the bottom of the visualization. To provide more information about the specific reads, we will zoom in until the specific nucleotides can be read. Sequence Read View. Clicking on a specific read brings up a pop-up window. This pop-up window displays information about the read. This includes an alignment between the read and the reference, as well as the chromosome, coordinates, read length, template length, cigar score, flag, and name. If the read is paired, the position of the other segment will be displayed below. Other sequence read features. Clicking on the More button and then Reference DNA Sequence brings up a panel that allows us to copy and paste the reference sequence that is present for our current visualization. Clicking on the More button and then one of the Highlight buttons will allow us to highlight the alignment using a mouse selection or chromosomal coordinates. Clicking on the Download GDC BAM Slice button will download the current view in BAM format. For more information on the GDC data portal, please refer to the other videos or go to docs.gdc.cancer.gov.